Okay, let's get started. Announcement, there is a chemistry seminar this afternoon about neuroactive peptides, small molecules. Should be interesting, but very small molecules. Who cares about small molecules? We just care about big molecules, right? So that's the only announcement I had as far as that's concerned. Wanted to review a little bit the Lac operon that we talked about last time. Lac operon is a really nice example of bacterial regulation that basically uses both kinds of regulation. Standard, the real kind of regulation, at least according to Jacob and Monod. The negative regulation with a repressor, but also having positive regulation with an activator. And here, repressor is bound in the absence of ligand, which is the lactose. Again, this is all about breaking down lactose. If you have lactose around, then you want to express these genes. If you don't have lactose around, you don't want to express these genes. But you only want to express the genes when you need glucose. So if you've got glucose around, there's no reason to make these genes. It's only in the absence of glucose and the presence of lactose that you can have this. So two signals coming in, presence of lactose, absence of glucose, and those then get integrated into giving you activation. And so this is the classic kind of case which we have with bacterial regulation. Binding sites for regulatory proteins either right next to where the polymerase is going to be binding or overlapping where the polymerase is going to bind. Of course, as always in biology, there are exceptions. And one of my favorite exceptions is this one here, which has to do with a bacterial transcriptional regulator that binds a long way away from the promoter that it's actually regulating. And this is the case of what's called NTRC, nitrogen regulatory protein C. That in and of itself is not that important. What is important here is that this is a slightly different kind of promoter in bacteria. So you've got a different sequence, which means it probably also has something else that's interesting about this particular polymerase. It's got a different sigma factor that's associated with it. And what this polymerase does is it sits here and does not transcribe. Unlike normally what you have with sigma 70 and normal bacterial RNA polymerases, this now can only transcribe if it has interactions with this activator protein, which is quite a long way away. And so what happens is there's this loop that forms between this protein bound, this sequence called an enhancer, and we'll get back to enhancers a little bit more later on, that then loops over, interacts with the polymerase, and now, after ATP hydrolysis, now you have helicase activity, the polymerase starts, etc. And this is actually seen in the electron microscope. This blob here is NTRC. This blob is RNA polymerase. And they interact here directly with this loop of DNA in between. This is from Sidney Kustu's lab, my PhD advisor at Berkeley, published in 1990, which actually right as I just got started working on this project. So this looping interaction is very transient. And by transient, I mean this happens very infrequently, and that is basically the subject of this. Which is, it is yellowed, yes. This is my PhD thesis. And down here at the bottom, Sidney Kustu, who was one of the chair of my thesis committee. And down at the very bottom it says 96, but we can ignore that for the time being. But the main thing that I showed there, again, 6 years of my life, was that this interaction was a very brief interaction. It was actually really hard to detect, which was a pain. Chapter 4 of this thesis. Yeah?
Is it caused by the enhancer or the promoter?
Pardon?
Is this caused by the enhancer or the promoter?
Okay, so the question, this is a great question. Is the looping caused by the enhancer or by the promoter? The answer is neither. It's a protein-protein interaction. So it's a protein-protein interaction between the enhancer binding protein, in this case NTRC, and the RNA polymerase sitting at the promoter. So it's a protein-protein interaction that gives you that looping interaction. And that brings us actually really nicely to the next slide. Thank you for the introduction, where I want to talk a little bit more about enhancers. So an enhancer is a DNA sequence. Oh, sorry, Ian.
So this kind of seems analogous, and maybe I'm drawing lines where I shouldn't. This kind of seems analogous to the activator protein binding to the enhancer in eukaryotic, and which then loops back and touches onto the mediator. Is there any relationship there?
Is there any relationship between this enhancer binding protein in bacteria and what's happening in eukaryotes? Great lead into this slide. Yeah, the answer is definitely yes. Or at least I try and convince people that that's the case because I was working with this bizarre enhancer binding protein in bacteria. Who cares about enhancer binding proteins in bacteria? We care about enhancer binding proteins in eukaryotes. Way more interesting. But it turns out enhancers serve in an almost identical way. And again, so this enhancer is a DNA sequence, and all that it does is serve as a place for enhancer binding proteins to bind. And those enhancer binding proteins are then transcriptional activators that can loop through protein-protein interactions with the pre-initiation complex, and it's not just the mediator. We'll get to that in just a second. So this idea of DNA looping and, again, just protein-protein interactions that's happening through space. And this is a really bizarre kind of thing, and people didn't think it happened. Again, the bacterial paradigm is everything sitting right next to your promoter. So it turns out you can put enhancers tens of thousands of nucleotides away from the promoter, and they'll still work as long as you have that enhancer binding protein. And it turns out that great reason to study the bacterial enhancer binding proteins is that it's a much simpler system. You're just talking about the polymerase as opposed to all this other mediator stuff, et cetera. But one of the big questions was, when this enhancer binding protein sits down on the DNA, does it then scoot along the DNA until it gets to the promoter and then help the interaction? Or is it literally one of these looping things? And so we saw the loops in the electron microscope. OK, that's nice, but anyone who's done electron microscopy knows there are lots of artifacts in electron microscopy. So you have to be very careful about it. So there was another experiment that was done also in the Cousteau lab where they use these things called catenanes. And we've talked about catenanes a very tiny little bit already. But what a catenane is two circles that are just linked to each other by the fact that they are circles and just interlinked with each other. There's no direct way you can have an enhancer bound on this circle and a promoter bound on this circle. If it zooms along the DNA, it's going to zoom around and around and around and never end up hitting that promoter. So there are tricks using topoisomerases to make this kind of construct. And if they're linked together like this, then you get activation. If they're not linked together, you don't get activation. So it's a really nice way of showing that this looping, what was seen in the electron microscope, is also really what's going on in terms of this activation process. And again, this is what I mean by the different DNA molecules down here at the bottom. Turns out that this looping DNA is flexible. I brought my model today, of course, but this is the non-flexible model. We've talked about DNA being flexible a bunch of times before, particularly if you think about nucleosomes, et cetera. But it's not infinitely flexible. And so it turns out if just by flopping around, you have a more or less optimal distance that you can have between your enhancer and your promoter. And it turns out that that one is just about 500 nucleotides. If it gets too far away, then it's harder to find. If it gets too close, because you can't infinitely bend the DNA, it ends up being less good. And so this is just an estimation of that. So if you look at right next to the promoter, sure, you can have protein-protein interactions. If you move it further away, the DNA can't bend very well. You move it far enough away, the DNA bends well. You move it further and further away, it's harder and harder to get those kinds of interactions taking place. And this is literally done with this kind of system here, looking at the bacterial one, later shown in the eukaryotic system. There's one other thing that I wanted to mention as far as bacterial transcriptional regulation is concerned, and this has to do with what I just talked about, this particular bizarre sigma factor right here that interacts with NTRC. It It's binding to a different promoter. So you've got a different sequence that is being bound to by the RNA polymerase. And in this particular case, the sigma 54 promoter, it all stays together. It's a closed complex, and you don't get that opening until you have the interaction with the enhancer binding protein and ATP hydrolysis that takes place. But this is also a nice way that bacteria can regulate their genes in a sequential fashion. So turns out bacterial viruses are really good at doing this, but it works for other bacterial systems as well. If you have a gene that encodes a sigma factor, it has to be transcribed before that sigma factor can take the RNA polymerase and direct it to different promoters, which can then express another sigma factor, which will cause that RNA polymerase to go to yet other promoters. And it turns out that one of these that was been really well studied turns out to be a sigma factor that's really important for expressing genes like HSP70 and HSP60 genes. So on heat shock, if you do heat shock to E. coli, you express this sigma factor. And that sigma factor is going to cause all the genes that have the appropriate promoter for that to be overexpressed. So it's just another way of doing gene regulation just by expressing a different sigma factor. So we have positive regulation, negative regulation, regulation at a distance with enhancer binding proteins, and different sigma factors. Yes, there are a few other ones, but these are really the main ones. Questions about bacterial transcriptional regulation? Yes?
Does it work the same way with enhancers being transcribed as well? Or are the enhancers?
Okay, so the question here has to do with enhancers. And okay, so this had the same thing to do with enhancers is basically the question. So it's true for enhancer binding proteins, yes, but the enhancer is a DNA sequence. And so that's just to be replicated with the rest of your DNA. But yeah, it's definitely true. You could have, for instance, one of these genes that's being expressed here turns out to be a transcriptional regulator, which can then be used for expressing other genes. And that's classic. That happens all the time, and not just in bacteria, but in eukaryotic systems as well. So one of the things that transcriptional regulators will regulate transcriptional regulators. And you end up with these sort of feedback loops and progressions that happen. Okay, now everybody's had a chance to get their clickers out? Good. So the lactose operon, now we talked about all of these different things here, is subject to negative transcriptional regulation, positive transcriptional regulation, regulation by DNA looping, both negative and positive regulation, regulation by alternative sigma factors. Still aiming for that 100%. Ten, 5, only 83 of you here today? That's not very many. Okay, so if we had added A and B and D together, we'd be at 99%. But it's it's negative and positive, and unfortunately there's no way to count both on your clickers here. So yes, it is both negative and positive regulation. Negative regulation due to what? All right. Well, what's the name of the protein? It's the lactose repressor, and the activator for positive transcription regulation is CAP-CRP. Okay, so now, finally, eukaryotic regulation. So this process here turns out to actually be really, really similar to what's going on with NTRC and enhancer binding proteins, but also the same thing is going on with lac, operon, et cetera. You've got interactions with gene regulatory proteins that are literally right next to the promoter, but you also have lots of here regulatory sequences that can be literally tens of thousands of base pairs away from the promoter. So that's what they call the gene control region. Can be tens of thousands of nucleotides bigger than the actual protein coding piece of your gene. And of course, that usually in eukaryotic cells has introns and exons, et cetera. How many of you remember how many protein coding genes there are in the human genome? Approximately. About 20, 25,000. There are a heck of a lot more promoters on here, right? 70-odd thousand promoters. So many, many more. And in fact, so I may get a chance to pull this up again. Just a couple of days ago, there was a paper talking about long non-coding RNAs, and I think they said there are about 50,000 promoters for just these RNAs that are not coding sequences. So lots and lots and lots of promoters. How do they know that these are promoters? By doing those chromatin immunoprecipitation experiments that we talked about last time, just looking at where the RNA polymerase is. RNA polymerase 2, where's it bound to? And looking at TF2D complexes. Where are these things bound? They're only bound at promoters. There's a whole bunch of them. Regulatory proteins, we mentioned this last time, that a bunch of genes in our genome encode these regulatory proteins, mostly enhancer binding proteins, but we'll also talk about some of the repressors and co-activators, co-repressors. But they're in very small numbers, which we also talked about last time. We talked about the purification processes. These affinity chromatographies, for instance, are really important for isolating these individual regulatory proteins because there are very, very few of them. And being able to study them means you need to get enough of them at all. So it's quite a challenging process. How do all of these things associate? Again, your gene regulatory region is tens of thousands of base pairs. It's all about the looping. And in many cases, the mediator as well. But that's not the only case. Of course, sometimes you have direct interactions right here with some of these gene regulatory proteins. Why should we care? Mentioned this also last time. Transcription factors, again, absolutely horrible name, but it's what's used all the time. I would say these are enhancer binding proteins as opposed to transcription factors. In 2009, it was shown that 4 of these, if you take them and express them in fibroblasts, which are pretty standard cells, those then become pluripotent stem cells and can develop into whole organisms, which is really pretty darn amazing. We talked about nanog last time. Nanog is one of these, originally 4, now they've gone down to 3 proteins that you can overexpress in some of these cells and get them to become pluripotent stem cells. Really amazing. And for this, the group of, I think it was Susumu Tonegawa? No, that was I'll try and remember the name. If I can't remember the name, I won't ask you to remember it on an exam. But his group got the Nobel Prize in literally 3 years after publishing this paper for showing that just the expression of these 4 proteins could convert completely differentiated cells into pluripotent stem cells. The other reason that we should care about these really low concentration DNA binding proteins is that there are hundreds of thousands of these, or more than 500,000 of these protein binding regions in the human genome. So over 8% of the human genome has binding sites for these proteins. And if you consider that 1.5% of the genome is protein coding, this is really an amazing amount. I'm still trying to figure out what most of these things are doing. It's great to have a catalog and say, oh, look, all these things are happening, but converting that, actually getting the phone numbers that go with this list of names is still a lot harder to do right now. So there's lots to do, which is great for all of us who are in the research area here. So what do some of these enhancer binding proteins look like? Here just called gene activator proteins. They always have 2 domains. Sometimes they've got more than 2 domains. But What would you expect one of these proteins to have? Have a DNA binding domain because it's a enhancer binding protein, enhancers DNA, and an activation domain. Turns out that these activation domains are really kind of unclear. They've got lots of different things that they have. DNA binding is pretty clear. We talked about DNA binding last time. But what do these activation domains look like? Basically, the take-home message is they're sticky. Why sticky? Well, because it's all about the protein-protein interactions. Your question before about whether it's the enhancer that's involved, it's really more about these protein-protein interactions. So it's that sticky part of your enhancer binding protein that's important for this. How does it become sticky? Acidic, proline-rich, glutamine-rich. Seems to be lots of different ways that you can have an activation domain. One of the really interesting things that was shown quite early on when people were looking at how these enhancer binding proteins were put together is that it turns out you can mix and match the domains of these various different proteins. Turns out to be ridiculously useful for looking at all kinds of protein-protein interactions and figuring out what's going on inside the cell. We'll talk about that hopefully when we talk about the methods right at the end. But basically, it's really just about getting that activation domain associated with the DNA at an enhancer that gives you the transcriptional activation. And so how that was shown is basically here. This is in yeast. The Gal4 protein, which is a enhancer binding protein, has an activation domain and has a DNA binding domain, interacts with things at the promoter, gives you transcription. Using fun molecular biology recombinant DNA techniques, you can take the Gal4 activation domain and stick it together with a different DNA binding domain that binds to a totally different sequence. And if you do that with either this gene here for the galactokinase, which is regulated by the Gal4 protein, or something that's being regulated by just this sequence. Here we have the LAC-Z gene. LAC-Z normally being regulated in bacteria by Repressor cap protein. It's really convenient. It's got this enzymatic activity that's not found in almost all eukaryotes. So if you put that LAC-Z gene into a eukaryote and then look for that enzymatic activity, it's only going to be making that enzymatic activity if it's being transcribed through this artificial piece of DNA which you put inside the cell. So this artificial piece of DNA with the LAC-Z gene, if it's got the DNA binding sequence that recognizes the Gal4 DNA binding domain, in this case, normal Gal4 protein, you're expressing LAC-Z. If ever you now switch the DNA binding sequence, change the enhancer sequence to a totally different one, but now can interact with the DNA binding domain that you've also put together with this extra protein, now you get gene activation. So the activation domain and the DNA binding domain are completely separate from each other. And you can do all of this mixing and matching and get them all to work. Also mentioned they thought this as being modular. So you've got a DNA binding module and a activation module. Those things can be put together. So what are they doing? Again, these are sticky. What are they doing when they're sticky? We already talked about interaction with the media. Oh, sorry, Ian, do you have a question before that?
So, just trying to wrap my head around it. Do you ever have the same activation protein or domain? You have that on different binding domains within the same organism?
Okay, so the question basically, sorry, I'll paraphrase this, is do you ever find one, just like a single activation domain with a whole bunch of different DNA binding domains in a normal organism that the molecular biologists haven't messed with? And the answer is no. You will find similar ones, similar activation domains and different DNA binding domains. And those may have been mixed and matched in evolution, but that's a much harder question to answer. But yeah, this is a completely artificial system that we're looking at here. And basically the question that excuse me, Mark Tashney, I think at Harvard, was trying to answer was, can you separate these things from each other? Can you separate activation from DNA binding? And so yes, in this case, you certainly can. Okay, so what are they interacting with? We talked about the mediator already. Here's our modular enhancer binding protein, some distance away from your promoter. The activation domain can interact with the mediator. That's why the mediator was named the mediator in the first place, was because it was mediating the interactions between these gene regulators and the RNA polymerase. But originally, interestingly enough, the interactions were shown between general transcription factors and these transcriptional regulators. The lab right downstairs from where I did my PhD was very much involved in looking at how these things were interacting with each other. And they found that they had lots of interactions with TF2D. Now remember, TF2D is made up of what? It's made up of TBP, the TATA binding protein, and so TAFs, the TATA binding protein associated factors. What do those TATA binding proteins associated factors interact with? Sometimes they interact with enhancers. And so that was one of the very first things to be shown in terms of how they were interacting. Then a competing lab actually showed that mediator was important for a lot of these things. And clearly the writers of this textbook, on one side of that divide versus the other side of the divide. So it's these interactions with mediator, general transcription factors. One thing I did also wanted to mention here is this, it's called the holoenzyme. Where did we talk about holoenzyme before? Sigma factor together with the core polymerase in bacteria. Here, for some promoters, you seem to have TF2D, TF2B, all of these TF2H, TF2F, all together as a holoenzyme, and that gets brought to the promoter. And so last time when we looked at, well, actually a couple times ago when we talked about transcriptional regulation, first it's B, so first it's D, then B, then E, then F, then H. In some cases it's just D and everything else. And that everything else is going to be your holoenzyme. So direct interactions between activators and some part of the pre-initiation complex is a common way that you can have regulation. But because eukaryotes are ridiculously complicated, we also have a number of other proteins that are involved in this whole process. And these are called either co-activators or co-repressors. A co-activator or co-repressor, the sort of definition of these proteins is that, yeah, they'll act as activators or repressors, but they don't bind to DNA by themselves. So just that protein by itself, a co-activator or a co-repressor, doesn't have any activity. It's got to somehow be brought to the DNA through protein-protein interactions, and then it can serve as an activator or repressor. And that's basically just shown down here. Multiple DNA binding proteins, these are the little guys here, 4 different little guys, binding to the DNA, and then interacting with either co-activators or co-repressors. And one of these things that sort of want to bear in mind here is that this green subunit here, this would be some enhancer binding protein binding to a particular sequence, but depending on who its partner is, it could be interacting with a co-repressor or a co-activator. So in this case, binding by this DNA binding protein to this DNA sequence doesn't tell you whether it's activating or repressing. It could be one or the other, depending on what the other partners are. And it's a combination of all of these things that leads to either repression or activation. So here, this green one can interact with co-repressors or co-activators. This red one can interact with co-repressors or co-activators. It's only red and green together that interact with co-repressors. And now these colors are not important here, but the idea is that it's this mixing and matching which can happen, and it makes it ridiculously complicated. And in fact, that's this paper that I refer to right here. You can take a look at it if you like. They've taken literally using that same modular fashion that I was talking about before. And again, an artificial system, but they know they've got about 400 different regulators and co-regulators that can all interact with each other in different ways and give different outcomes. So trying to wrap your brain around this is rather complicated. Trying to write decent exam questions about it is also really complicated. So I wanted to also bring up a couple of other ways about looking at this process. These are not figures from the textbook, but they're things that I find make it a little bit easier for me to think about. This is a nice review paper. You can access it down here, PubMed Central 2833331. I wanted to put it up there so people can take a look at it later. Sorry, this isn't in the notes that were posted online, but be up here. But basically, all of these are possible kinds of interactions we can have here. You can have different DNA binding proteins right next to where the promoter is. You can have DNA binding proteins in enhancers. Of course, here everything's associated with chromatin. These can interact directly with RNA polymerase II and the general transcription factors with our nice little TATA binding protein here. Or they can be interacting through co-regulators. Those co-regulators could be interacting directly with the RNA polymerase, but also, as we'll talk about toward the end of today's lecture, interacting with the histones, because histone association with the DNA is a big problem. You can't get this whole RNA polymerase II pre-initiation complex to a promoter if it's really heavily associated with the nucleosomes. And it's even hard to get some of these DNA binding proteins associated with the DNA. We have the breathing that we talked about last time, but if it's a piece of DNA, it's right in the middle of a nucleosome, these interactions are not going to happen very frequently. So another figure from that paper, and basically everything you need to know about transcriptional regulation or initiation regulation in eukaryotes is here. But basic message, you start out with a chromatin template with all of these nucleosomes associated with the DNA. You need to get an activator somehow in there. And the reason for getting the activator in and bound to DNA is it's a lot easier to get that relatively small activator complex associated with the DNA than your whole ginormous, probably holoenzyme, which has almost 100 different proteins that are associated with it. So getting that single activator protein associated is very important. Then you get the polymerase. The polymerase gets associated here, starts doing abortive initiation. You get phosphorylation, eventually move to elongation, go up to termination, and this whole cycle goes around and around. Each of these steps, 1, 2, up to 8, are all being regulated. We're not going to talk about all the different kinds of regulations that's happening there. But the real key here is getting your first protein associated with this particular piece of DNA. There's another thing which turns out to be very useful in terms of putting together these pre-initiation complex. I mentioned right at the beginning the studies done with NTRC and bacterial enhancer binding proteins were showing that 500 nucleotides away is just about right. Further away still works. But you can actually get a little bit closer if you have a specific DNA bending protein. And very often these DNA bending proteins are really important for pulling all of the complexes together. And we already talked about this with TBP. TBP bends the DNA, and in bending the DNA allows the various different TAFs in particular to associate with the different parts of the promoter. So it's this bending process that can help the enhancerosome. Again, horrible use of zomes. It's a nucleic acid plus proteins. And so your nucleic acid is particularly the enhancer, sometimes your bending sequence, and then the enhancer binding proteins and all of the co-regulators that are associated with it. Yeah?
So the enhancerosome is the sequence that it binds to as well as the proteins that are bound to it?
Yes. Anytime you have zome, sorry, just to repeat this, anytime you have zome, that's going to be proteins and DNA. So the ribosome is nucleotides in DNA. It's the RNA plus the proteins. The nucleosome is the DNA plus the histones. So spliceosome, it's going to be your RNA plus the proteins. So it's always going to be nucleic acid plus proteins. So enhancersome. And the main thing here is it's really a structure, lots of proteins, a reasonable amount of DNA, which is associated here. And then each of these colored fragments here is going to be serving as an enhancer with these various different enhancer binding proteins. You also can have some co-regulators here, like this one that's not binding to the DNA. And that structure is what's interacting with the RNA polymerase. So what are some of these co-activators, co-regulators here? Well, because you've got a nucleosome-bound template that the RNA polymerase is supposed to be binding to and transcribing, not surprisingly, you need to modify the chromatin structure. We'll talk more about modifying the chromatin structure a little bit later on, specifically how it happens with some of the co-activators and co-repressors. But it turns out the RNA polymerase itself actually also modifies nucleosomes. The names of the proteins here are not critical, so I won't talk about them. They're associated, however, with what? RNA polymerase II. Where do you put these extra proteins that you might need? Hang them off the CTD, exactly. And so what these proteins do is they modify histones as you are undergoing transcription. And what they do is they will acetylate histones as the polymerase is moving forward and methylate them after it's come through. And so this guy is a histone acetyltransferase, puts acetyl groups on. This guy is a histone methyltransferase and adds all of these methyl groups at the end. And basically what that does, it loosens up the chromatin in front of your polymerase and tightens it down after the polymerase has come through. And so it's giving you a separated polymerase. Once these are methylated, it's actually a lot harder to get transcriptional initiation to come through here. So you've started transcription, you'll make that gene once, but you won't make it a whole bunch of other times. So that was just one thing to mention. This is mentioned in the text, but I didn't have a figure for it, so I pulled this figure off online. So there's, however, a big problem that you have when you've got all this looping, again, tens of thousands of nucleotides, even different pieces of DNA. If you've got an enhancer binding protein, how does that enhancer binding protein know what promoter to interact with? Because if it's interacting through space, just through looping and protein-protein interactions, they've all got a mediator. They've all got an RNA polymerase too. So if you're going to use this looping way of doing activation, you have to have some way of protecting against this looping to the wrong place. And the way that works is through insulator elements. And anytime you talk about these elements, what does element mean? So elements are not proteins. Elements are always going to be your nucleic acid. So an insulator is a nucleic acid, just like an enhancer is a nucleic acid. So these insulator elements are nucleic acid sequences that do exactly like what we talked about. They insulate whatever's between these two insulator elements from this enhancer binding protein going off and doing the wrong thing, activating the wrong gene. So these regulatory sequences are going to be in the same part of your genome with the gene that they're actually regulating. How does this work? Not surprisingly, there are insulator binding proteins that will isolate these loops of DNA sequence between these insulator elements. And when we talked about transcription, we were looking at DNA and puffs in chromosomes or looped domains that you had in the lampbrush chromosomes. That's exactly what's making these loops or puffs. It's the insulators, the insulator binding proteins, which are isolating these particular sets of DNA so you can have regulation happening just on those sequences. Yeah?
I thought that the activation domain on the activator protein, I thought that that was specific to the gene.
No. So to paraphrase Ian's question here, he thought that the activation domain on your individual enhancer binding proteins can be specific to any given gene. Well, what that activation domain is doing is it's interacting with parts of the general transcription machinery. Could be mediator, could be any of the general transcription factors. Those are the same for every single promoter. So these are not specific per genes. And so, right, because it could interact with any of these genes willy-nilly, all over the place. And so you have to have these insulators in order to separate it. Now, how you can separate this guy, now how these insulator binding sequences actually work in space, literally in the nucleus, is still a pretty open question. And we're not entirely sure exactly how that works. Yeah?
Do we know how the insulator binding proteins decide which
Yeah, you can get them out.
to loop together and fold? Like, how do we stop the insulator element that's supposed to be separating gene A and B from binding to the one that's supposed to be separating gene N?
Right. So the question here is, this is a great question, is how do the insulator binding proteins know where to bind? And the answer to that is we don't really know. But what seems to happen is it's the two insulator sequences which are closest to each other. So that's what seems to happen. But again, this is an open question. And we'll actually talk about this more later when we talk about some more regulation that's going on later on. So the other thing that I wanted to mention here is I mentioned sort of puffs, and these insulator domains are very often next to those puffs or the loops that you see in the lamp brush chromosomes. You can actually identify those if you take an antibody to those insulator binding proteins and hybridize it with a chromosome, and that's what's shown down here. All the green stuff are your insulator binding proteins, and the red is just these sort of puffed regions. So look, we've got two nice insulators here around this puffed region on the outside, etc. So you can see them spread out through the genome. We've been trying to localize, people who work on this, try and localize the genes that are separated into these two, and they do seem to be co-regulated. You can identify those particular genes which are going on there. So everyone saw that there's a clicker question on the next slide, so I don't need to remind you again. Which of the following are the most likely reason that insulators are required? DNA can bend, heterochromatin, euchromatin, enhancers, or mediator? Ten. 3, 2, 1. So most people seem to think it's enhancers. Some people think it's DNA bending, and we've got a few votes for those other answers in there as well. So why do you need an insulator? It's so that the enhancer binding protein doesn't go and interact with the wrong genes. And so why would that be? It's because of the fact you have enhancers. That DNA can bend, yeah, but DNA can bend doesn't really make a difference whether you have an insulator or not. So, and we can also have various different, pieces of DNA which can serve as enhancers and other places. Yeah?
Can you explain the difference between
Hetero and euchromatin. We're actually getting to that in just a second. So we'll go back to that. Now there is one other thing that let's get rid of these. Here, one of the things that I forgot to mention is these other kinds of sequences that we talked about before. Remember barrier sequences? Barrier sequences? When did he talk about those? We're talking about chromatin and modifications to chromatin and the spreading of those chromatin modifications and the whole feedback loop. Once you make a change, all the changes are going to happen there. So the barrier sequences actually act in not dissimilar ways to these insulator sequences. They're a DNA sequence that you have a specific protein that binds to, sometimes nuclear matrix, some kind of enzyme, etc. So they're functioning in a pretty similar way, except they're blocking the progress of either euchromatin or heterochromatin. So getting back to your question, euchromatin versus heterochromatin. So euchromatin is what? It's the decondensed chromatin. It's the true chromatin, if you want to go back and look at the origin of the word. But it's really about being available for transcription, being available to get binding of your regulatory proteins, binding of the RNA polymerase to that. And here, nice example is right in the middle here, where you've got loosened up chromatin. Heterochromatin, on the other hand, is the compacted chromatin that's really been, compacted in such a way to shut down any kind of transcription from there, but also just compacting your genome, because compaction of the genome is really important in order to fit everything into into your nucleus. So here, thinking about gene regulation, you can have your gene activator protein, again, interacting directly with the pre-initiation complex, which is great and wonderful. But for the most part, that's not what the DNA looks like. It's usually at least packed in chromatin and very often packed in heterochromatin as well. So after you have binding by your gene activator protein, somehow you've got to get the pre-initiation complex associated with this TATA box and start transcription from happening. So very often these gene activator proteins will also interact with chromatin remodeling complexes, and that's this little piece sticking out here, the protein-protein interaction, also to show this is a way bigger complex than your standard gene activation protein, and that can just move nucleosomes around in such a way that hopefully will give you access to the TATA box, this little green thing right here. Very often, once you have chromatin remodeling, and we looked at this before, remember, chromatin remodeling is ATP dependent. All of these are ATP dependent processes. Can also bring in histone chaperones that'll just take the nucleosome off completely. That's great, because then you can access your TATA box very easily. And in some cases, these histone chaperones will exchange the individual histones. And it turns out that some of those histones, those alternative histones, are very prevalent right at where you get transcriptional activation. And it turns out that those histones, which are really good at helping to bring general transcription factors into the DNA and get them to the right place. And then the other set of proteins that are involved in how you get from just your activator protein to access to the TATA box are histone modifying enzymes, so specific modifications to the side chains of your histones. And we talked about that really briefly in terms of acetylation, methylation, etc. Particularly here, acetylation of your histones is going to loosen them up so that you can have interactions with these general transcription factors. And again, these are the TF, A, B, C, D, etc. Yeah?
These happen one or the other, they don't all happen, right?
So, question is, do you have interactions with the chromatin remodeling complexes or interactions with the histone modification enzymes? And the answer is, you've got a whole bunch of them happening at different times at different genes. So sometimes, and also this is the oversimplification here, is it's one gene activator protein. It's almost never one gene activator protein. It's usually multiple gene activator proteins interacting with these co-regulators And very often the co-regulators are chromatin remodeling complexes or histone modification enzymes. So it's lots of different things, and usually at one individual promoter, you'll have multiple gene regulatory proteins interacting with multiple of these co-regulators. So one of the best studied of these systems has to do with, two different kinds of regulators. I don't expect you to remember all the details here, but this is described for one particular set of chromatin remodeling and histone chaperone proteins, SWI/SNF, which remodel your histones in such a way that the activator can now bind. Again, these are ATP-dependent processes. And then once you have remodeling, you could either have just this activator bound to the DNA, you can have the histones be completely removed, that's what we had here, or changing out the various histones in such a way that this is now a positive way that you can have a specific nucleosome that's going to help transcription taking place at that place. So this is another sort of cartoon where sort of kind of beating the dead horse here. Lots of different ways to have transcriptional activation. Here is just with one particular transcriptional activator. It's our little green one here. Very often that's going to help another transcriptional activator, in this case a specific transcriptional activator. It can interact directly with different parts of the polymerase. Again, all of this is getting your RNA polymerase to the promoter. Once you're at the promoter, if TF2H activity is there, then you'll have transcription. But in many cases, you actually need a different activator protein to now stimulate transcription from going from there. And that's maybe, Ian, remembering the cartoon that we actually, was more the animation, where you've got everything sitting together at the promoter and then waiting for that transcriptional activator to come in. And so that would be this model here. And in some cases, the RNA polymerase goes a little way away from the promoter, starts to elongate, but gets stuck at this point. And then sometimes these transcriptional regulators can help move it along from there. So lots of different things that these transcriptional regulators can be doing, and usually lots of interacting ones as well. One way of looking at this is looking at one particular nucleosome. This is an example now looking at the specific modifications that happen on these nucleosomes. You have a transcriptional regulator that binds to this little dark green sequence here, and then histone acetyltransferases that interact with it. So here's your green transcription activator, your histone acetyltransferase. It's your co-activator, which will acetylate histones. Once you have histone acetylation, that changes the structure of the tail because you've got change of the charge interactions which you have there. In this particular case, you also have a histone kinase. What do kinases do? Put phosphates onto your histones. Phosphates have a nice negative charge, so they're also helping to push away the DNA from this nucleosome. But it turns out that TF2D, one of those other TAFs, actually has a domain that interacts with phosphorylated histone side chains. So a specifically modified histone side chain helps bring TF2D into this complex. Some of the other modifications are recognized by different chromatin remodeling domains and, chromatin remodeling complexes. In those are various different domains. There are two sort of flavors of domains of these histone modification enzymes. Get to you in just a sec. There are the so-called bromodomains and chromodomains. I can never remember bromodomain, but chromodomains are associated with heterochromatin. What do we usually see? Anyone remember heterochromatin? What's usually happening with heterochromatin in terms of modifications in histone side chains? Usually methylation. And so it turns out that chromodomains really like to bind to methylated histone side chains. And the other kind, the bromodomains, are binding to those acetylated side chains. in particular. So yes, your question.
So up there it shows the histone acetyltransferase putting the H3K9
Mhm.
and the histone kinase adding the phosphate, but it's happening on both sides. Are there transcription activations on both sides that are adding it to both of those side chains of the histone, or is the one side transcription activator adding it to both sides?
Oh, so the modifications that are happening there, and so yes, showing the phosphorylation that's happening on the other side, as well as the change in the methylation, which is going on here. It seems that this modification here, these tails flap around a lot. If you think about the nucleosome, it's kind of this disc structure. And so probably the single transcriptional activator interacting with the kinase can modify all of those side chains. Plus, of course, there are multiple nucleosomes all right next to each other, so it's going to be reaching over to the next one as well. Okay, so chromodomains and bromodomains. Again, bromodomains, acetylated histone side chains, chromodomains, methylated histone side chains. So I've mentioned a lot, 2 different transcriptional regulators, and in fact, usually a lot more than 2 different transcriptional regulators that are associating with the DNA. And one of the things that was also found through some of these experiments with mixing and matching your DNA binding sequences and activation domains, even with actually just exactly the same activation domain, if you bring 2 of those activation domains together onto the DNA versus one or the other, they're really synergistic in terms of how they activate DNA. So if you've got one activator that's going to give you a small amount of transcription, another activator that gives you a small amount of transcription just by themselves, if you put the 2 of them together, you get way, way, way more. So this process of having multiple regulators gets us back to what we talked about in terms of cooperative binding before, really giving you much more of an on-off switch in terms of how different genes are being made, as opposed to a little bit, then a little bit more, then a little bit more. And so it does seem that we've evolved all of these processes in order to have much more of a switch-like regulation rather than, anybody know what a rheostat is? Some of you, not enough of you. More like a dial in terms of the different amounts. There are some cases, but for the most part, it's going to be an on or off, or almost all on or almost all off kinds of situation. And this, I think, is getting back to your question about different genes and different regulations that's going on here. These are multiple different steps that you can have in activation. And this goes all the way back to when we were talking about that extra image that I brought in, 1 through 8, in terms of various different regulation steps. So each of these different steps here can be going on at any given promoter. So almost always you're going to have a gene activator protein first that's going to interact with your chromatin. How you get to the chromatin could be breathing, could be some other kind of modification that's happening first. But once you have that first interaction, again, it's usually a relatively small sequence, you can get that gene activator protein in. Very often it'll be other activator proteins that can come and associate there either directly or very often with these chromatin remodeling complexes and histone modification. Then you can get all of these extra other activator proteins. They can either be your direct DNA binding or co-activator proteins, and many of these are histone modification enzymes or chromatin remodeling complexes. And then you can finally get your holoenzyme, everything put together here. And the big thing here really has to do with the size. If you just think about all the stuff you've got in your holoenzyme, mediator, general transcription factors, RNA polymerase, this thing is huge relative to that chromatin-bound template that you're talking about in the beginning. And then once this is made, you have to have a couple of other things that will happen in order to actually get elongation to take place. So this is an overall kind of picture, but just in general, it's that binding of that gene regulatory protein, which is eventually going to lead to, through all these extra steps, finally getting transcriptional initiation. And we talked about transcriptional initiation way back at the beginning. Once you have transcription started in this elongating polymerase, which is ridiculously processive, it's going to burn through a lot of NTPs until it finally gets to the end of your particular gene. So it's very important to regulate this. And once you have made that decision, that RNA is going to get processed, it's going to get made into protein, etc. So regulation early on in that first transcription step is the really, really important one. And that's why we're sort of spending so much time talking about this. Okay. What's he going to do now? Clicker questions. I got you well trained. Which of the following seems to be the rate-limiting step for eukaryotic transcriptional inactivation? Enhancer binding protein binds to an enhancer, covalent histone modification, mediator DNA binding, RNA polymerase DNA binding, or TF2H activation? Please talk, please interact. That's the idea. Ten, 5, 3, 2, 1. Okay, we seem to be about half and half split between enhancer binding protein binding to the enhancer or covalent histone modification. What's an enhancer binding protein interacting with an enhancer? It's an activator protein interacting with the DNA. And so that happens because you got your little thin space that it needs to go into. The covalent histone modification almost always is happening later. Chromatin remodeling you could argue about. But the histone modification, how is that histone modification going to happen? You have an interaction with your enhancer binding protein. So it's that protein-protein interaction which is there. So A is correct. I want to spend the last couple of minutes talking about repressors. Repressors are almost exactly the flip side of your enhancer binding protein. They bind to a silencer. Now, I don't like the repressor binding site. I like the silencer binding site because these can act just in very similar ways to you have enhancers. They can be upstream, downstream, tens of thousands of nucleotides away, etc. Sometimes these sites are actually going to overlap with enhancers. So an enhancer and a silencer are going to overlap with each other. So it just depends on which protein you have. If you've got more of this protein than you have of that protein, or this protein binds better than that protein, it's going to block the activator binding, no DNA binding, no activation taking place. Sometimes you've got, we just talked about the sticky part of the activation domain. Well, that could stick to one of these silencer binding domains. So that will block any other kind of interactions. And sometimes these guys actually will interact directly with general transcription factors and block whatever interaction these activators would have with these general transcription factors. And this will now block the association of all the rest of your general transcription factors together. So TF2D bound to DNA, if there's one of these silencer binding proteins that are associated with it, that will block any of the other proteins getting into the complex. You can also have indirect interactions, and this is just the flip side of those histone modifying enzymes. If you have an histone acetyltransferase, that's going to lead to Euchromatin formation and activation of transcription. On the other hand, if you have a histone methyltransferase, it's going to methylate those side chains and lead to compaction. So these silencer binding proteins are going to interact with the histone methyltransferases, stimulating methylation taking place. So this is just a figure showing some of these interactions. This is a paper from a little while ago. And if any of you have a chance, this is a really funny takeoff on All About the Base. that one of my buddies at Baylor College of Medicine put together. If you need a study break, I think it's about 4 or 5 minutes. Hilarious. Have a good weekend.